So next up, we have Dr. Madeleine Cule. She's a director of data science at Calico Life Sciences and uses statistical modeling to better understand the interplay between genetics, environment, and age in driving disease risk using data from large human cohort studies. The ultimate goal is to generate new therapeutic hypotheses for aging-related disease. And she is going to be telling us about deep phenotyping in the UK Biobank for Aging Research.
So it's absolutely fantastic to be here today and share a little bit of the work I and my colleagues have been doing on using large human cohorts to try to understand the aging process, changes that happen with age, and how they relate to disease. So for a computational scientist, it's just a fantastic time to be alive with the advances in AI, machine learning, and also really, really interesting data sets that we can use to ask and hopefully answer questions that we've never answered before. So just to get started, I'll tell you a little bit about Calico, for those who aren't familiar with this company. This is a company that was founded by Alphabet just around 11 years ago. And the mission is to harness advanced technologies and model systems to increase our understanding of the biology that controls human aging, and then ultimately to use that to devise interventions that enable people to live longer and healthier lives. So I'm going to talk a little bit about my personal favorite model system for human aging, which is humans. As I mentioned, that's been an area where there's really been a complete revolution in the past 10, 15 years. So as I think everyone here knows, aging is a main risk factor for the most prevalent disease. And starting in early adulthood, the chance of death due to the most common causes increases exponentially, which is shown in this graph with age on the x-axis and chance of mortality per disease. And on the right, this kind of aggregates that into all-cause mortality. So this makes the aging process a really interesting thing to understand if we want to try and treat disease. And so biobank scale data on humans represents an enormous opportunity for research and discovery in this field. So rather than studying individual cohorts that focus on a specific disease, we now have access to resources at population scale, meaning hundreds of thousands or even millions of participants who've been deeply profiled with prospective health follow-up. And this is well powered for multiple disease endpoints. And this is a really great study design for an aging company, because rather than just looking at diseases in isolation, we can look potentially at changes that happen during aging that affect multiple disease outcomes all at once, and sort of not miss anything, and also potentially make new discoveries that are new connections that weren't known before. So as part of amassing these biobanks, people were recruited without any ascertainment by disease status and subjected to comprehensive measures that varies a little bit between studies, including potentially biomarker measurements, functional measurements, some imaging data, again, all across the body to give a window into changes that occur before the start of disease. And this gives us an opportunity to have an unbiased look at the health of all across the body and how these are connected over time as participants age. So I've just illustrated here with the slide from the UK Biobank, which recruited 500,000 people, to illustrate some of the measures that were measured, including biomarkers, cognitive measures, questionnaires about lifestyle, physical measurements, genetics, which we'll talk a bit about later, and all linked to health outcomes which are collected over time. So really interesting resource for us. I'm particularly going to talk today about the new opportunities that are emerging thanks to the collection of imaging data. So beginning back in 2014, tens of thousands of volunteers in this data set were recruited for really detailed imaging. I've illustrated some of the modalities here across the body, vascular system, bones, brain, and heart. And this gives a real opportunity to try out the latest techniques in machine learning to extract new measurements that might give us understanding of disease outcomes. And then just to talk a little bit about how this fits into a framework for advancing drug discovery. We kind of use these measurements that we make in humans to identify new age-related traits that might have a heritable component. and then use molecular data, which is again being collected at an amazing rate, to interrogate causal relationships at the molecular level, and then use these to as a platform to develop new therapeutic hypotheses that we can then test either in independent human cohorts, like experimental medicine approach, or experiments in mice, in vitro systems, a whole lot of opportunities that we'll potentially hear about from other people later today. So I'm going to talk about a particular example of the work that we've been doing that uses deep learning to extract information from abdominal imaging in the UK Biobank. So this is a data set that was collected that I've illustrated on the left that contains really rich information about body composition and organ health linked to multiple aging-related diseases, cardiovascular disease, cancer, cardiometabolic disease. And when we started this work about 4 or 5 years ago, this was really underexplored in population-level studies. The data had been collected, but no one had really figured out a good way to analyze it. The brain and cardiac imaging was a little bit more mature. Brain doesn't move around, so that kind of helps. The tools there were pretty matured. But the abdominal imaging was relatively underexplored. And there's a large degree of variation between individuals, so registration-based approaches hadn't been that successful. So we teamed up with a team of radiologists to annotate individual organs within this data set, and then worked with machine learning engineers to train a neural network that could sort of segment out individual organs and structures across the whole data set, and use this to create a platform for discovery based on imaging-based traits. So this annotation process was pretty time-consuming, so we limited this to just 100 individuals, capturing a range of age, sexes, and body shape and size. So we started with just a few organs in this original paper, but have now captured more than 80 traits derived from body imaging. And then using this relatively small data set, we could use pretty standard machine learning architecture to extract all these abdominal organs and then apply this across the whole data set of tens of thousands of individuals. And so after we've done this process, we now have a library of precise quantitative measurements on around 80 IDPs or imaging-derived phenotypes like organ volume, dimensions, blood vessel measurements, fat quantification, and also measurements of iron. And now we've applied this to a library of 80,000 participants. So this is a really interesting data set to tell us about aging-related disease. So that's and then just to sort of highlight what these measurements are telling us, we can use these, thanks to the kind of linkage with health outcomes, we can look in a very unbiased way across the whole disease phenome to try to understand connections with different diseases. So I've illustrated this here with these volcano plots for each of a few traits, fat and iron measurements, and volumes of things like visceral fat and so on, and shown that these are linked with organ-specific diseases, which seems kind of obvious now, but at the time it really wasn't clear what these traits might tell us. So this tells us that we're getting kind of, even in a non-disease cohort, we can pick up changes that are predictive of future disease outcomes. And then thanks to the study design of the UK Biobank, we were able to conduct genetic association studies and identify a number of loci associated with these traits that are also associated with disease. And this is pretty routine now to calculate complex traits and use genetic studies to look at the relationship with disease outcomes, but this is something that wasn't really possible before the collection of these huge biobank data sets. Just to give a couple of examples of new discoveries that come out of this type of analysis. One of the traits that we looked at that hadn't been studied in population cohorts was iron in the liver and spleen. And this was something that we think is quite interesting related to aging because iron homeostasis or loss of iron homeostasis plays a major role in many diseases of aging that's really kind of poorly understood. And our genome-wide association study here, we didn't really know what to expect, but we thought it was kind of interesting that this picked up genes that were associated with erythrocyte structure, and also this one interesting locus called MS4A7, which is an aging biomarker that had been linked to immune changes in aging, and this was associated with iron in the spleen, and we think this might be associated with how macrophages engulf dying red cells and could potentially be an important aging marker. And again, we can integrate molecular data with this data set to try to understand these loci in more detail and potentially try to understand how they're related to aging-related disease. Another example of the type of trait that we looked at in this data set is the cardiovascular system. So I've illustrated this on the, can you see that? Yes, oh, that's nice. On the left, here's again the body scan, and we've looked at, segmented out the vascular system and the heart, and then looked at the diameter or the health of the blood vessels at different anatomical locations, so starting at the aortic arch here and then down the aorta. And this gives us a way to look at aneurysm or aging of the large blood vessels in a way that isn't really accessible using disease, using just clinical information. So I just show this as an example of how we can use this imaging data and machine learning to extract traits in an otherwise healthy population to study aging-related changes that aren't often really very well captured by the medical system for things like aneurysm or changes in iron accumulation that might be occurring before the start of disease. So in the last few minutes, I'm just going to talk about two future directions that we're thinking will be to talk about two future directions of how we're planning to develop this work to better understand the aging process. In the work that I've just described, we really focus the machine learning work on replicating what a human would do or what a human would think is interesting from an image. But one of the advantages of the kind of latest machine learning models is we don't really need to anchor ourselves to what a radiologist might see or a radiologist might think is interesting. So I've been working with a team of machine learning engineers who've been, rather than trying to replicate particular manually derived features, have instead been using representation learning-based approaches to just summarize the whole image or a particular organ into a vector that just describes the image but isn't necessarily human interpretable. Then explore various ways to turn these into phenotypes, for example, by building a model that associates it with a disease outcome. And then this gives us new measures that we can use to try and look at links to aging-related disease. So this is just one example. We've classified based on a kind of disease label for osteoporosis, looking just at the pelvis in these images, and then have classified, this just gives two examples of extreme low and high disease burden. And then in a genome-wide association study, this picked up kind of information that kind of made sense. So genes related to osteogenesis, which kind of makes sense for osteoporosis risk. Finally, what I described so far isn't necessarily specific to the aging process. So starting in about 2020, Calico teamed up with CCI and the UK Biobank and the MRC in the UK to expand this study and the data collection to include a longitudinal component, so inviting these tens of thousands of individuals back to repeat the imaging process. And this, I think, is a really exciting extension for the perspective of aging because it allows us to characterize aging-related decline across a huge range of imaging phenotypes, to identify potentially traits where a change is predictive of disease outcomes, to characterize genes really involved in the aging process, and identify biomarkers of disease progression, which could be really important for future clinical trials. So this is a data collection that's ongoing that we think is really going to unlock a huge range of opportunities that we don't even know about yet. I'd like to end by acknowledging a fantastic team of collaborators, both at Calico and at the University of Westminster and Lincoln in the UK, who we work with on their adiposity and abdominal IDP. As I said at the beginning, for a computational scientist interested in health and disease, this is just an amazing time to be working on this kind of work, and I'm really excited to see what ideas the whole field can come up with for exploring this data set in the future. So it's been a great opportunity to give you a flavor of the kind of work we're thinking at Calico, and I'm looking forward to the rest of the meeting. And I'd be happy to take some questions or have some discussion in the next few minutes. Thank you.
One question I had is just sort of like natural variation within the population based on body sizes, like ethnic background and so forth, like ancestry. Like how much noise were you seeing in the MRI imaging data just due to that? Or I guess one question in addition to that is like, how might these diseases look different on MRI based on those backgrounds, right? Your starting point in organ volume or subcutaneous fat, something like that.
So I guess I'm not sure if I completely understand the question. Are you asking about the kind of technical aspects of how do the models
do we have
handle
enough
the
data?
variation?
Yeah, do we have enough data for that among enough different groups yet? Or is this like a huge gap where we actually should be going out and recruiting more of like a certain ancestry and so forth?
Yeah. So I guess two parts to that question. One is kind of technical. How did we make sure this worked across the whole population? And this was kind of an iterative process where we started with annotating 100 individuals. And then we chose those to cover a range of ages, body size, just based on kind of basic demographics. And then as we looked at the output of the model, we'd kind of look at extremes in those axes. And if we found it wasn't performing well in a particular particular types of data or particular areas of the data set, we could go back and add more annotations until we got a good performance. So that was a kind of semi-automated approach, and so that was something that was more important for some traits than others. On the kind of representativeness of the data, I think that's super important for the genetic studies in particular. And the UK Biobank is mostly, it's recruited in the UK, so there's a mix of ancestries, but it's kind of representative of that population. Tends to be kind of healthier and whiter than the population as a whole in the UK. And this is something that, particularly with genetic studies, the field is kind of working towards, but I would say we're not there yet. And with the imaging, it's a little bit further behind the genetic studies. One opportunity that I didn't talk about, sorry, last thought on this, then you have time for another question. Last thought on this is we've also got a separate project looking at using clinical data that was collected not in this way, but just trying to use that, and that's been pretty successful, and that could be a way to get access to data on a broader range of individuals using clinical radiology. Thank you. Yeah.
Hi, amazing talk. So I just have a curiosity. So longitudinal studies are important for the predictive power. So having like a scan or whatever biomarker taken like at regular interval is gonna help also to monitor kinetics, because different people may have different environmental exposure. So of course you are relying on the UK Biobank, which I think they just started collecting this data, but are you also planning to build a model when you are gonna use like measurement? Because with the biomarker of aging, we are trying to build like a biobank program, and one of the goals is actually taking measurements every year. So is this something that you're planning to do in the future?
I think that probably won't get everything from one single study design. So I think you're talking about maybe a smaller cohort that's most densely sampled, and then the challenge will be integrating information from all these different data sources. I think the clinical, kind of getting into the clinical data that's already collected, that's just lying around in hospitals, is also a really nice opportunity to have more densely sampled data. Yeah.
Thank you.
Okay. Building on that, I'm just curious if you have done any comparison about what type of drugs or class of drugs that might lead to specific phenotypes. If you're using like anti-inflammatory or specific IL-23 or IL-24, and if you see specific changes that are maybe not expected or are interesting to lead to one phenotype or the other.
That's a really good question. I haven't looked at that specifically in in this UK Biobank, although we do have self-reported information on what drugs people are taking. A little bit harder to interpret because it also tells you about what can just be a biomarker of the disease process. But that data is there. Just I think the analysis would need to be a little bit framed carefully to interpret it well. Yeah.
Thanks, Madeleine, for the great talk. Just out of curiosity, do you see any value in approaching it from the angle of, I know you're selecting the IDPs based on healthy and broad range of population, but what about taking it to the extreme of looking at the disease, like particular diseases, and going and building that data set from there?
I think that's one thing we're quite excited about with the representation learning approach, specifically using disease labels to try to look at how does this disease manifest in the imaging. And then that can potentially give you a way to get undiagnosed cases, just people who kind of look similar to people who have that diagnosis, because we kind of take the labels as absolute, but really if you think about how the process goes to how a diagnosis gets to the clinical record, that's not always the case. So that's one approach that we think has quite a lot of mileage left to go. Thank you very much.